Yes. So, very good evening, students. Yeah. So today we're going to start with the topic called photosynthesis again, and it's going to be a revised topic. Revised topic. Why? Because আমরা লাস্ট উইকে যা করেছিলাম সেটার একটুখানি রিক্যাপিচুলেশন করে we are going to start. ফাইন, ঠিক আছে? তো আমি ধরেই নিচ্ছি তোদের কাছে আগের দিনের যা পড়েছিলাম সেটা অতটা হয়তো মাথায় আছে কিনা নেই, but still let me start with a little bit of recapitulation and then we are going to get into the next level. Fine. So the topic is about photosynthesis again. It goes it is very evident যে এই টপিকটা খুব important topic. So this topic is very important topic. The topic is about photosynthesis. কতগুলো questions আমরা আশা করতে পারি in NEET from this topic? We can expect not less than 2 to 3 questions. Fine. So let us talk about photosynthesis. So a basic idea, ছোট্ট করে first 10 minutes. Photosynthesis means what? Synthesis means formation of something in presence of light. So what is that something? Normally carbon dioxide atmospheric carbon dioxide is being fixed in presence of 12 molecules of water and in presence of sunlight and chlorophyll. In presence of sunlight and chlorophyll, tell me what is being made? Glucose, C6H12O6 plus 6O2 plus 6H2O. So over here what we are finding out that this particular oxygen is liberating out. This oxygen comes out by the oxidation of or the photolysis of water. This is very clear to us. তাই তো জলটা ভেঙে by splitting of water we ultimately get what? We ultimately get oxygen. So this water is being oxidized. The process is called oxidation. The water is being oxidized and this carbon dioxide is being reduced. This carbon dioxide is being reduced to give rise to what? Give rise to glucose. So in carbon dioxide we have got carbon and oxygen. So carbon and oxygen. What is being added? Hydrogen is being added. What is being added? Hydrogen is being added. And this addition of hydrogen is called what? Addition of hydrogen is called reduction. It's called reduction. So this carbon dioxide is being reduced. This carbon dioxide is being reduced. So a chemical equation, a chemical reaction where oxidation and reduction takes place simultaneously, we call it redox reaction. So this is photosynthesis, which is an anabolic process. What is anabolic process? The process where we get something which is constructive. And by this process we have been able to make glucose, and this glucose is called as the respiratory fuel. This glucose is called as the respiratory fuel of life. That means every individual, প্রত্যেকটা individual, every life, not only individual, every life, be it from bacteria to a dinosaur, that means a unicellular prokaryote to a multicellular large organism, eukaryotes, all we need is what? Is respiratory fuel called glucose. Glucose ভেঙে আমরা কি পাবো? By breaking down glucose we can get what? We can get energy. So by this process, the glucose in presence of oxygen being broken down to give rise to carbon dioxide plus 6 molecules of water plus 686 kilocalorie of energy it comes out, which is as good as 38 ATP molecules. Fine. So by breaking down one molecule of glucose, what we can get is we can get 38 ATP molecules. Any issues about that? So this is called as the respiratory fuel of life, and we know about ATP. ATP is called as what? The energy currency of the cell. So every other cell needs ATP to run their normal metabolism of the body. হয়েছে না হয়নি? Great. তাহলে এই পার্টিকুলার প্রসেসের জন্য আমাদের কি জানতে হবে? What we need to understand, what are the requirement? The requirement that we have already talked about is sunlight, and the total sunlight's total wavelength that is required for photosynthesis is called as PAR. What is the full form of that? Photosynthetically active radiation. Full form ta ki? Photosynthetically active radiation. So this ranges from 400 to 700 nanometer. 400 to 700 nanometer. And what percentage of the total sunlight is being accepted by the green plants for making the food? Photosynthesis korar jonne? Yes. what is the percentage of the sunlight which is required or which is absorbed by the green plants? It is total in world 2 to 10% of the total solar energy is utilized by the green plants to make the food. And again, we need the visible wavelength of light. We need the visible wavelength of light. Now, we need the visible wavelength of light. And what is the visible length? We know that it comes where? Between 400 to 700 nanometer. And remember, by the help of the experimentation done by T.W. Engelmann, if you remember that, T.W. Engelmann, what he did, he took one prism, allowed the sunlight to pass through the prism, and he kept what over here? In the test tube, he kept cladophora. Ki rekhechilo? Cladophora rekhechilo. And found that this light is going to get split into different spectrums called VIBGYOR, and ultimately what he saw that the aerobic bacteria, and because more of oxygen evolved in the blue light and more of oxygen evolved in the red light. From here, what we can get is the action spectrum. Ki pelam amra ekhan theke? Action spectrum. And what is action spectrum? The spectrum where the maximum what? Photosynthesis occurs. And what is that light? Blue and red. Now, what is there inside this cladophora? What is there inside cladophora? Arju, tell me, what is so very important about cladophora? What is cladophora? It's a green algae. And what makes it green? Chlorophyll. That means this is the pigment which is the next requirement. This is the next requirement. So sunlight is required. Next, pigment is required. So when we talk about pigment, tokhoni mathaয় আসছে, sir, pigment ক রকমের হয়? How many types of pigments are there? Ankita, is this clear? Now, so when we talk about chlorophyll, we have got an idea that chlorophyll, in plant kingdom we have studied about chlorophyll A, chlorophyll B, chlorophyll C, chlorophyll D. Fine, we have talked about them. Now, chlorophyll is a pigment. Chlorophyll is what? It's a pigment. Now, let us get into a little bit basics about this chlorophyll. Now, when we talk about chlorophyll A is the main one which is present as a reaction center. It acts as a reaction center. Along with the reaction center, there are some other components which we call it as antenna. And what are those antenna? There are three of them which helps in making the antenna. Thus, chlorophyll B acts as antenna. Carotenoids acts as antenna. The next one is xanthophylls. Last day in the class we have talked about the pigment's color. This carotenoids, we know beta carotene present inside carrot. And what is the color of it? Red to orange. And when we talk about xanthophylls, the color of that becomes yellow. Xanthophylls. And one of them can also become, some of them can also have a brown type of thing. And one of the xanthophylls that we have studied in plant kingdom under brown algae is fucoxanthin. So, what is the reaction center and the function of antenna? Do you know function technique? Reaction center is always chlorophyll A. This is the main reaction center. And these antenna, and the composition of antenna is what? Mainly carotenoids, xanthophylls, and chlorophyll B. So chlorophyll B is not the reaction center. Mind you what? Now, think about, this is the main synapthy. Who is the synapthy? The reaction center is the synapthy. Chlorophyll A. Bolo ke guard dicche kara guard dicche. Who are next to this? The next is what? Chlorophyll B, carotenoids, and come on, and xanthophylls. Got the point? All of them together makes what? Especially these antenna makes what? LHC. What is LHC? So antenna are also called as LHC. What is the meaning of that? Light harvesting complex. We call it light harvesting complex. So these antenna, they can take some lower wavelength than 680. 680 or 700. So, say for example, the wavelength of light coming up as 400. So 400 nanometer, 400 nanometer. The moment this particular wavelength comes over here, it's having what? Low the wavelength, the energy is inversely proportional to lambda. Fine? Energy is inversely proportional to the wavelength. So if it is having low of wavelength, it is having high energy. It's having high energy. So what it's going to do is, the moment this gets excited, this 400 is going to give the energy to say 450 nanometer. It's going to give the energy to say 480 nanometer, more than that. And it's going to give the energy to what? It's going to give the energy from high energy to low energy, because it's having what? More of? More of lambda. More of lambda means less of what? Less of E. So it is having high of E, high of energy. It's going to give the, got the point? Give this particular. So the wavelength of light, these accessory or antenna, they are going to activate what? Ultimately it's going to activate into 5820, then it's going to activate 580, and then something like, it's going to activate this one as 680 nanometer. Got the idea? 680 nanometer. And what do we know about 680 nanometer? Last day we talked about this. 680 nanometer is what? P680, that we call it as PS2, photosystem 2. So we call this as photosystem 2. So photosystem 2 gets activated. Do you think it can get activated alone, or it needs the help of LHC, light harvesting complex? অনেকগুলো মিলে কি করলো? It's activated what? The P680. একা কিন্তু P680 straight away, it never got excited. Got the point? The moment P680 gets excited, it is going to emit what? It's going to release electron. It's going to release electron, and this electron will be accepted by what? Some electron acceptor. This electron will be accepted by electron acceptor. We call it primary electron acceptor. And tell me, last day's class, what we have talked about? What is the primary electron acceptor of PS2? It is pheophytin. It is pheophytin. Is the primary electron acceptor. Is going to accept the electron. Got the point? So always remember, any wavelength of light, if it is less than 680 nanometer, if it is less than 680 nanometer, then PS2, that is P680, is going to get activated. Also there is another photosystem called photosystem 1, which gets activated at 700 nanometer. The 700 nanometer or 680 nanometer tolai, if there is a less than 680 nanometer, these two of the photosystem gets activated. Again I repeat, photosystem 2 and photosystem 1, both of them gets activated if the wavelength of light is less than 680 nanometer. Now Ankita is going to tell me, if the wavelength of light has become 690 nanometer, tell me out of these two, which is going to get activated? Is this that both of them can also get activated? Only PS1 gets activated. Only PS1 gets activated. Because 680 এর উপরে চলে গেছে 680 nanometer, PS2 কিন্তু অ্যাক্টিভেট হবে না। মাইন্ড ইউ, এইখানেই ম্যাক্সিমাম স্টুডেন্টের প্রবলেম আছে। যদি এই জায়গাটা তোরা বুঝে যাস, তোদের ফটোসিনথেসিসের সাইক্লিক অ্যান্ড নন সাইক্লিক ফটোফসফোরাইলেশন নিয়ে কোনো প্রবলেম হবে না। এইটু ক্লিয়ার হয়েছে? ইউ টেল মি again, if this is the wavelength of light which comes over here, name the photosystem which gets activated. Option A PS2, option B PS1, option C both of them, option D none of them. Both of them. I said less than 680 nanometer হলে তাহলে দুজনেই অ্যাক্টিভেট হয়ে যাবে, and দুজনেই ইলেকট্রন ফ্লো করাবে। And mind you, PS1 का जो प्राइमरी इलेक्ट्रॉन एक्सेप्टर है वो डिफरेंट है। वो क्या है? FES is the primary electron acceptor from PS1. কোনো অসুবিধা আছে? And mind you, every other term I'm talking about is some of the answer to some of the MCQ, previously asked question paper. Clear? So what is the primary electron acceptor of PS1? FES. What is the primary electron acceptor of PS2? Pheophytin. Okay, done? When they both of them going to get activated? When the wavelength of light is less than 680 or more than 680? Less than 680. That is very, very important. Are you clear? Now, what is the location of this PS1 and PS2? What is the location of this PS1 and PS2? The next one is very, very important. What is the location of this PS1 and PS2? What is it? Okay, let's make it like this. Okay. This is outer membrane, this is inner membrane. What we are making right now? Chloroplast, okay? Chloroplast. This is outer membrane, inner membrane. Ankita, name two cell organelle having double membrane. And chloroplast, mitochondria and chloroplast. So this is the outer membrane, this is the inner membrane. Fine. Now, inside what is present? Inside what is present? Come on. What are these? What are these? Grana. What are these? Grana thylakoid. Remember? Last week we talked about these are granum. These are thylakoids, and many of the thylakoids together makes what? Granum, where the light reaction takes place. And there is another one. There is a stromal lamellae. There is a stromal lamellae or stromal lamellae by which two of the granum gets connected. By which the two of the granum gets connected. Fine. And you know what? Where is the location of PS2? The PS2 is present over here on the granal thylakoid. On the granal thylakoid. Let me write over here. PS2 present where? On the granal thylakoid. Granal thylakoid. On the granal thylakoid. What is the location of PS1? The location of PS1 is present over here. This is PS1. This is PS1. So what is the position of PS1? What is the location of it? They are again present where? In the granal thylakoid. In the granal thylakoid. Are you getting me? Any issues? But what is the location of PS1 again? The location of PS1 is again present over here on the stromal lamellae. This is what? This is stromal lamellae. This is stromal lamellae. Okay? And what is this? Outside granum, what is present? Stroma. Outside granum, what is present? Stroma. Clear? Now, what is this one? This one is PS2. What is this one? PS1. What is this one? PS1. Clear? Now, let us have a look on this particular event. I'm just showing you this particular event. So PS2 and PS1, both of them. PS2, PS1, both of them are present on granal thylakoid. PS1 only present on stromal lamellae. Clear? Any issues with that? And we also call it as stromal thylakoid. We can also call it as stromal thylakoid. Let's get into this. Okay. This one is, come on, this one is PS2, this one is PS1, PS1, this is okay, this is P680 nanometer, that means PS2. This is what? P700, that means PS1. This is PS1 and this is PS2. And this is the membrane. And this is the membrane. This is the membrane. Okay, this is the membrane. What is this? Granal thylakoid. Granal thylakoid. So it is present on the granal thylakoid. Okay. What is outside? Stroma organa. Very good. Stroma. So what is present outside? Stroma. Very good. Matrix. Now, if the wavelength of light is less than 680 nanometer, less than activated, PS1 gets activated, electron starts emitting, fine, and accepted by, help me, phyophytin, over here, FES. Very good, FES. Now you guide me, FES. From FES this electron is going to come to FD. What is FD? FD is what? Ferredoxin. So this electron now has come down to phaedoxin. From phaedoxin, this electron comes straight away to NADP plus 2H plus 2 electron. This electron is going to make it what? Reduce it with to NADPS2 by the help of the enzyme NADP reductase. NADP reductase. Ankita, tell me, the formation of NADPS2 has formed inside the granule thylakoid or outside the granule thylakoid inside stroma? कहाँ पे बना? Mind you, this is an answer to a question. NEET question. NADPS2 কোথায় তৈরি হয়? The portion where NADPS2 gets formed is where? Inside granule thylakoid or outside granule thylakoid inside stroma? Clear? Inside stroma. দেখো এটা stroma মধ্যে আছে তো? Okay, done. What is the location of NADP reductase enzyme? Stroma, grana, where? Stroma, sir. Fine. Ultimately क्या बन गया? NADPS2. NADPS2 बन गया. अच्छा, Ankita, you tell me, this NADP has been formed basically in presence of light or in absence of light? So can we call this as an event of light reaction, light dependent reaction? तो अगर light reaction में NADPS2 बना क्यों? Sir, dark reaction में चाहिए sir. This is needed in dark reaction. That's why it is being produced. Dark reaction এ NADPS2 লাগবে। এই জন্য এটা তৈরি হয়ে বসে আছে। কোনো অসুবিধা আছে এখানে? Any issues? আয়ুষী? চলো, very good. Now, what is the fate of this electron? Phosphatidine নিয়ে কি বসে থাকবে? No. It's going to give the electron back. So it's an uphill. Now it's going to come down, called downhill. Fine? এই ডাউনহিলটা এইভাবে নামবে, আমি এইভাবে না দেখালে পারতাম। Fine. So who is going to take it? First, PQ. Tell me, what is PQ? Plastoquinone. What is PQ? What is PQ? It is plastoquinone. Plastoquinone. And from plastoquinone, this electron is going to come over here to cytochrome. Cytochrome B6F complex. Cytochrome B6F complex. This electron is going to come from here downhill towards another acceptor called plastocyanin, PC. So what is PC? PC is plastocyanin. বল এই তিনটেকে কি বললাম? কি বললাম? Come on, from phosphatidine downhill এ কি আছে? প্রথমে আছে plastocyanin, তারপরে আসছে cytochrome B6F complex, and then electron comes back to plastoquinone. Plastocyanin. So plastocyanin. This electron comes back to PS1. Fine? Clear? হয়েছে? During this flow of electron from one acceptor to the other, during the flow of this electron, a slightest amount of energy can come out, is released. This is energy which is released. This is energy which is released. This is energy which is released. Clear? This is E which has been released from here. And this energy are required for what? This energy are required to make what? ATP. ADP plus IP is going to make what? ATP. Is going to make what? ATP. Now this ATP which is being formed by this electron flow, this whole downhill concept is called ETC or ETS. What is that? Electron transport chain or electron transport system. Got the idea? Open your book. If you have got old book, page 233. Page 233, electron transport chain or electron transport system. Or if you have got new book, it is 160. Just check it out. Yes. This is the electron transport system. Which is very, very important. Clear? Over here, mitochondria is being shown over there. The blue colored key in a blue colored mitochondrial membrane. Okay? Now, all of you, wait. Just have a look. Blue color বাদিকে আর ডান দিকে কি আছে? Left and right, what is present? What is present in the left and right? Huh? Inner membrane space and matrix. Inner membrane space and matrix. For let me give you this idea. This is mitochondria, and this is what? Tell me, inner membrane. And you can see the inner membrane is having more surface area. And in lower classes we know that these are cristae. These are between outer membrane and inner membrane. Can you see some gap? And what is present in this gap? What is present in this gap is H plus ion. What is present in this gap is H plus ion. And this H plus ion is being stored over here between the outer membrane and inner membrane. And what is this? This is called matrix. Eta kire? Eta matrix. Tale, if this is mitochondria, you can see the hydrogen gradient is being made. A proton gradient. A proton gradient is being. Yes. So, yeah. Sure. So there is a proton gradient which is made in mitochondrial matrix, between outer membrane and inner membrane. Now what is the location? That location ta kothay? Location of PS2 and PS1 present over here. And this particular process takes place over here. What is this process called? This looks like what? This looks like Z scheme. What is this called? Z scheme. The same thing we have seen in where? Granule thylakoid. The same thing we can see in the granule thylakoid. So this H plus ion can only get inside matrix through this opening. And what is this opening called? F0F1 particle. Opening ke ki bola hoy? F0F1 particle. We are talking about what? Mitochondria, chloroplast now. Mitochondria. What happens in chloroplast? The same thing happens, but ekhantai amra okhane CF0 আর CF1 bolbo. Ki bolbo? The opening as CF0 and CF1. That means over here H plus ion is going to get concentrated over here. And then H plus ion ke ana hobe kothay? In the matrix. In the matrix. And this H plus ion can move out through CF0 and CF1. You want to see over there? Check it out. Boy ta ber kor. Boy, go to the yes, next page. Next page. Yeah, page number 141. Have a look. Can you see the same figure being given? Yes? Can you see CF0 and CF1? Where from the H plus ion coming out? Show me the book. What you can see over here? This is the granule thylakoid. This is the matrix. H plus ion present over here. This H plus ion can move out through CF0 and CF1 and forming what? Forming ATP. Forming what? Forming ATP. Now, when this H plus ion starts coming down through H0F1 particle over here, it activates one enzyme called ATP synthase. Yes? ATP synthase. ATP synthase. Okay. ATP synthase. Okay. So this is ATP synthase. ATP synthase gets activated. ADP plus IP makes what? Makes ATP. And this happens when the H plus ion gets in through the clear. Any issues? আমরা আরো ডেপথে যাব, কোনো চিন্তা নেই। ব্যাপারটা বুঝলি, mitochondria and chloroplast, both of them, what they can make is by this particular process. See, by this particular process of ETS, ultimately what is the end result? ATP and NADPH2. Both of them, ATP, NADPH2 And what is the product? O2. So during this process, see over here, the electron is moving in this direction, and the ultimate fate of electron is by making what? NADPH2. এটাকে কি বলবি? Cyclic or non-cyclic photophosphorylation? Non-cyclic. Why you are calling it non-cyclic? Because the electron is not coming back to PS2 again. ये electron यहाँ पे back करके यहाँ पे नहीं आ रहा है. But mind you, at P680, if the electron final fate is what? Making NADPH2. But this P680, what it can do is it can break down water. That's what we call it as photolysis of water. কি করতে পারে? জলকে ভাঙতে পারে. It can break down water. It can break down water at the wavelength what? P680. This water in presence of sunlight can break down into H plus OH minus ion. Or we can also rewrite this as H2O in presence of light can give rise to half O2 plus 2H plus 2 electron. So this is the mechanism by which you can have huge amount of H plus ion over here. Clear? Now O2 is being released. This O2 comes over here. This O2 comes over here. See, what is it? Stromal level. So this is PS2, PS1, PS2, PS1, and what is this? PS1. So the oxygen comes over here. So what is the net result? ATP, NADPH2, and oxygen. This is the result of light reaction. This is the result of light reaction. If this is the end product of light reaction, এগুলোই কোথায় লাগবে? ATP and NADPH2 are required where? In the dark reaction. বল এইটুকু জায়গা ক্লিয়ার আছে? কোনো অসুবিধা আছে বল? Is this clear? Got the idea? Now there is a term called Acha, first let me get into this is what? This is non-cyclic. This is what? Non-cyclic. If water gets split, then ultimately what is going to happen? Water is going to only split at 680 nanometer. Arjit bolbe. When it was less than 680 nanometer, name the photosystem which got activated. Both of them. But exactly at 680 nanometer, what is going to get split? Water gets split. তার আগে অবধি কি ইলেকট্রন বেরোচ্ছিল তো? Less than 680 or exactly at 680? Less than 680. So when it gets 680, তখনই কি হবে? ইলেকট্রনটা ফ্লো করে যাবে, and then this particular electron will be now huge because now water is now the source of what? Water is now the source of electron. Any issues about that? বাবা বুঝলাম না। তাহলে এই ইলেকট্রনটার ফাইনাল ফেটটা কোথায়? ফাইনাল ফেট হচ্ছে পেজ 233, ফাইনাল ফেট হচ্ছে পেজ 160. That complex 4A in electron transport system, you can see the reverse reaction. ওই পেজে দেখ complex 4A, complex 4 in ETS. What you can see is the final fate of this electron in the chapter respiration in plants. You can easily see half of O2 plus 2H plus 2 electron in complex 4, which we call it as terminal oxidation. কি বলি আমরা এটাকে? আমরা এটাকে বলি টার্মিনাল অক্সিডেশন। আর কি হচ্ছে দেখ, কি তৈরি হয়েছে? জল তৈরি হয়েছে। ওয়াটার ইজ বিং প্রডিউসড। What has been produced? Water has been produced. Got the point? Photosynthesis and respiration, both of them are interdependent. ভাঙলাম কোথায় জলটা? 680 nanometer. কি রিলিজ করলাম? What we have been able to release? Electron. So what is the source of electron? Water. And the same water is being formed in respiration, and that is why it is called terminal oxidation. And this is electron transport system. In electron transport system, this is the statement being given. এখানে কি লাইট লাগছে? Is light is required for this process? No. Got the point? Now what is non-cyclic? What is non-cyclic photophosphorylation? What is non-cyclic photophosphorylation? What is non-cyclic photophosphorylation? See over here. This is again PS2 first. PS2 first. This is what? PS1. Tell me the name of those. Tell me the name of this. Pheophytin. Then plastoquinone. Then cytochrome B6F. Then plastocyanin. Okay. This only gets activated at 700 nanometer or more than. Tell me the condition when PS1 gets activated. Ritam bolbe. Only when this is more than 680 nanometer. More than 680 nanometer, this is not going to. This is going to stop working. Who's going to work only? PS1 is going to start working. PS1 gets activated at 7 nanometer. এখান থেকে ইলেকট্রন চলে যাবে কোথায়? What is the acceptor? Come on, help me. FES. And this FES, if it moves the electron to FD, then what it's going to make? It's going to make NADPS2. বল বললি না 2H plus 2 electron নিয়ে কি করবে? NADPS2 তৈরি করবে. It's going to make NADPS2. But this is not going to happen. This electron is never comes back to FD. Fed oxygen কাছে আসে না during cyclic. So during cyclic, what happens? This electron, it goes back over here to the PQ. This FES gives back the electron to PQ, and PQ again starts forming. Ritam bolbe. The moment PQ gets electron, PQ is happy. The moment PQ is happy, come on, PQ is happy. PQ is happy. Receiving the electron. And when PQ is happy, it's going to make what? It's going to start making downhill. Tell me, downhill में बनता क्या है? ATP बनता है. What happens only in downhill? Only ATP gets formed. ATP plus IP makes what? ATP. So during cyclic photophosphorylation, what is the product?
ATP.
Only ATP. There is no formation of NADPS2. And this is a $1 million question. Mind you, you're going to get stuck when the question comes. So, মনে হয় না আটকাবে আজকে পরে. Come on. Got the idea? So, ইলেকট্রনটা কোন দিকে গেল না? FD কাছে গেলে কি তৈরি হয়ে যেত আবার? ওদিকে না গিয়ে FES থেকে কোথায় চলে গেল? PQ তে। PQ থেকে আবার স্টার্ট করলো এই জার্নিটা ডাউনহিল, আর ডাউনহিল নামলেই ও কি তৈরি করবে? Ankita, is this clear? Very good. Tell me, very good question. Very good question. It's a $1 million question. You should be awarded for this question. Can you see this? Tell me. When PS1 is going to work, if the wavelength of light comes more than 680, then only you are going to have PS1 working. But if it is less than 680, both of them are going to work. Kire, bhule jas na. Got the point, Ankita? We started the same thing over here. देखो तो यहाँ पे कुछ लिखा हुआ था, देखो तो. Less than? तब क्या हो गया? PS2, PS1, both of them happily enjoyed the process. ও এদিকে ATP বানাচ্ছে, ওদিকে NADPS বানাচ্ছে, ও বলছে দেখ আমি ATP বানিয়েছি, বলছে দেখ আমি NADPS বানিয়েছি. But if it goes more than 680, then 680 is not going to work. PS2 is not going to work. Only PS1 is going to work. और PS1 जब काम करेगा अकेला, तो वो किसको देगा इलेक्ट्रॉन? बोलो. It's going to give the electron straight away in this direction, not in this direction. It's going to again give it back to PQ. So PQ says the electron is going to come and ultimately make what? It's going to come downhill. It's going to show electron transport system, and ultimately what is going to form? Only ATP is going to form. একটু থাকবে বল বুঝলাম না What is ATP? What is the difference in Adrian? What is the difference between ATP and NADPS2? Which one is cash? Which one is check?
ATP is cash and NADPS2 is.
So if you want immediate energy, which should take place? Which process should take place? And you need to have that as a storage. Then you have to have check. That is NADPS2. Ball, bulgan. It all depends upon what kind of wavelength is coming up. Any issues with this? Kono doubt থাকলে জিজ্ঞেস করতে পারিস বিশাল ফাইন এনি ইস্যুস চল আমি এগোবো अंकिता बोलो समझ में आया अब बोलो साइक्लिक और नॉन साइक्लिक डिफरेंस थोड़ा देख लेते हैं साइक्लिक है ना ये क्वेश्चन आएगा आपके एग्जाम में द डिफरेंस बिटवीन राइट डाउन द डिफरेंस बिटवीन साइक्लिक एंड नॉन साइक्लिक फॉर फॉस्फोराइलेशन PS1 and PS2 difference between PS1 and PS2 cyclic and non cyclic let us have this idea चलो ना देख लेते हैं इसको. See over here in cyclic. In cyclic, क्या है P700? Electron is accelerated by FES. FES यहाँ पे लिखा नहीं है. It comes back to what? Electron कौन ले लिया इसको? ETS में कौन लेता है इसको? PQ लेता है. तो अल्टीमेटली क्या बनेगा? इलेक्ट्रॉन ट्रांसपोर्ट सिस्टम में तो ही है कि बनता क्या है? ETS से सिर्फ ATP बनता है, बाबा NADP नहीं बनता है. This is cyclic. This is cyclic. And see, this one is non-cyclic. See, this one is non-cyclic. In non-cyclic, देखो क्या हुआ? ये PS2 है, वाटर टूट गया. अच्छा, जॉल भांगे in presence of what? Mind you one thing. In presence of, again, very very important, Mn+2 and Cl minus. Mn+2 and Cl minus. Very very important. Mn+2 and Cl minus. Water splits. Photolysis of water হয় কার প্রেজেন্সে? Mn+2 manganese plus and chloride ion. Okay, now that जाने this is LS2. देखो चला गया and then it comes back during this process forms what? ATP. Again it comes back over here. The final state electron makes what? NADPS2. This is non-cyclic. This is non-cyclic because this electron never comes back to LHC2. That is photosystem to याद back करे ना. Clear এটা? अच्छा, into the agata. Yes, I want to show you the difference between PS1 and PS2. That এটা দেখাতে চেয়েছিলাম. See. Huh? Light P680. See, hydrogen acceptor, plastoquinone, cytochrome B6F, plastocyanin. Ultimately, it goes to what? P700. FNR is a reductase enzyme. It forms what? NADPS2. This takes place where? Inside stroma. Inside stroma. But inside this matrix, oxygen gets formed. Fine? And see, there is a pool of H plus ion. There is a pool of H plus ion. This H plus ion inside the lumen, there is a high H plus ion. It can only go outside, that is to a stroma, by CF0 and CF1. F0, F1 particle. Come on. And when it goes back, it makes what? It makes what? It activates an enzyme. What is the enzyme? ATP synthase. Enzyme name ki? ATP synthase. And makes what? ATP. This is the process by which ATP is being formed. And this process is called chemiosmosis. This process is called chemiosmosis. I'm going to start with the chemiosmosis. আমি এটা বলবো তার আগে আমি এটা দেখিয়ে নিই. PS1, PS2. Okay, it's not there. চল ঠিক আছে, নেক্সট একটা রয়েছে. আচ্ছা, now what is chemiosmosis? What is chemiosmosis? আচ্ছা, ইয়াটা clear? This is clear? Clear with you? চলো next. What is chemiosmosis? What is chemiosmosis? Acha, last day we talked about light reaction, है ना? We have talked about light reaction. We have completed light reaction. 2 steps we have talked about. Light number. Huh? Light reaction. In your exam, how you are going to write it? Light reaction. It is also called as what? Also called as Hill reaction. Also called as Hill reaction. Also called as Hill reaction. First step of light reaction. Achha, what is the site? Ayushi, tell me. What is the site where it takes place? Granum. Okay? The first step is what? Photolysis of water. Photolysis of water. The first step is photolysis of water. What is the meaning of the term photo? Photo means what? Light. Lysis means breakdown. So breakdown of light, what? Light in presence of water, or better of water in presence of light? Better of water in presence of light. So water gets split in light. And you tell me the wavelength? 680 nanometer. And for that, what is required? Mn plus 2 and Cl minus and Cl minus. Okay, chloride. And ultimately it's going to make what? Half of O2 plus 2H plus 2 electron. So electron is the this water is the source of electron. Acha. This Mn+2 and Cl minus, it helps the chlorophyll to remain intact. It stops photooxidation. লাইটের প্রেজেন্সে ওর ক্যারোটিনয়েডস যাতে ভেঙে না যায় ওটাকে মেনটেন করে, নাহলে কি হবে? ওই টার্মটাকে কি বলা হয়? ফটো অক্সিডেশন। Half O2 plus 2H plus 2 electron. Ultimately, we got O2. Fine. We can also write this as like this. See, in presence of sunlight, it forms H plus H minus ion. Such 4OH minus ion can bind in presence of light to give rise to 2 molecules of water plus oxygen gets evolved. Oxygen gets evolved. So this is a byproduct of light reaction. This is a byproduct of light reaction. Bushlam? The first step. What is the second step? The second step of light reaction is called photophosphorylation. Photophosphorylation. Shun means what? Shun means process. Ryle means addition. Addition. Phospho means phosphate. Phosphate. And photo means light. So the process of addition of phosphate with something in presence of light. What is that something? The addition of inorganic phosphate in presence of light with what? Adenosine diphosphate. It makes what? It makes ATP. This is the first process. The second what we can get is NADP binds to 2H plus. Again in presence of light to give rise to what? NADPH2. So NADPH and ATP, both of them are called as assimilatory power. Both of them are called assimilatory power. And this is the main product, ATP and NADPH2. Both of them are the main product. Era ki re? Era hocche main product. Ankita, tell me, what is the byproduct of light reaction? What is the byproduct of light reaction? O2 and water. O2 and water. But what is the main product? ATP and NADPH2. Bas, very good. So this is a light reaction. Okay, you got the idea what is photophosphorylation? Now who is going to tell me? There are two terms. One is substrate level phosphorylation and the one is oxidative phosphorylation. কে বলতে পারবি?
Sir, when only ATP is produced, it's substrate photosynthesis.
Very good.
And when no NADP and
No. Phosphorylation means what? Addition of phosphate with ADP to make what? ATP. So তুই বানাচ্ছিস কি? You are making ATP only. That is cash or check. Ready-made cash. What is the check? NADPH2. Okay, NADPH2 কে ভাঙতে গেলে তোকে কোথায় যেতে হবে? ব্যাংকে যেতে হবে। And tell me the name of the bank. Electron Transport System, Punjab National Bank, PNB, ETS. Okay, talk about that. তাহলে NADPH2 কে ভেঙে আমরা কি পেতে পারি? One NADPH2, when you break it down, this NADPH has been formed. इससे अगर आपको कैश चाहिए, ATP चाहिए, तो कहाँ पे जाना पड़ेगा आपको? Electron Transport System पे. So if you make ATP by the help of electron transport system, and where the starting point is NADPH2 or FADS2, then it is called oxidative phosphorylation. Oxidative phosphorylation. মনে থাকবে, আর সাবস্ট্রেট লেভেল ফসফোরাইলেশন কাকে বলা হয়? যখন ডাইরেক্টলি এটিপি তৈরি হয়। ডাইরেক্টলি এটিপি কোনটায় তৈরি হচ্ছে? কেমি অসমোসিস। এই দেখ এখানে यहाँ पे क्या बन रहा है? Chemi osmosis, or by the process of ETS. Chemi osmosis, when the energy which comes out, it helps in chemi osmosis, then only you are going to get what? Direct ATP, and that is what is called phosphorylation. What? Substrate level phosphorylation. কি বলা হয়? Substrate level phosphorylation. Vishal, is this clear? And what is oxidative phosphorylation? When you get ATP, but by breaking down NADPS2 and FADS2, where inside electron transport system. আমি আসছি ওটা পরে, বাবা এটা ক্লিয়ার? চলো নেক্সট। Chemiosmosis. I want to show you this. Chemiosmosis. Chemiosmosis. Fine. Chemiosmosis. Now this term chemiosmosis was given by Peter Michel. And he got Nobel Prize. What is chemiosmosis? Huh? This is a dam. Have you seen hydroelectric project? Dam দেখেছিস তোরা? জলের ড্যাম দেখেছিস না? Hydroelectric project যখন তৈরি করা হয়, টারবাইন থাকে। So this is turbine, okay? This is turbine, and this is the lock gate. This is the lock gate which can be open and closed. And this is the water body. This is the water body. এই water bodyটা এসে এখানে you are making what? You are making this. This is the dam. You're not allowing the water to come over in this side. So what you are maintaining? mgh. So what you are maintaining is potential energy. You're not allowing this water to go off in this path. কি রে বুঝলাম হয়েছে? This say it's for the sake of understanding. This is Bangladesh and this is India, okay? And this is Arijit. This is Arijit standing at the top of this dam, okay, with a bucket. কি রে বালতি নিয়ে দাঁড়িয়ে আছে। আর ভেতর দিয়ে কি আছে? ভেতর দিয়ে সিঁড়ি আছে, whatever. And অরিজিৎ কি করছে? এখানে অল্পটুকু জল আছে, আগেবার ছেড়েছিল একটুখানি, অল্পটুকু জল আছে। In this side बहुत कम पानी है। ये क्या था बाबू? टरबाइन। अंकिता बोलो टरबाइन का काम क्या है? ये लॉक गेट खुलेगा, पानी आएगा यहाँ पे, पोटेंशियल एनर्जी चेंज हो जाएगा इनटू काइनेटिक एनर्जी हाफ एम स्क्वायर किरे, and the whole city is going to get lit up. Electricity পেয়ে যাবি তোরা, হ্যাঁ? You are changing potential energy into kinetic energy. What is the term we are understanding? Chemiosmosis. And for osmosis, what is required? Gradient is required. So you have made a gradient of H plus ion over here. So what you have made? Gradient of H plus ion. And for that, what Arijit is doing? Arijit এক বালতি নিয়ে এই পাশে এসে এই পাশে গেছে, গিয়ে এক বালতি জল ভরেছে, ভরে আবার সিঁড়ি দিয়ে উঠে, থিঙ্ক অ্যাবাউট হাইট হচ্ছে প্রায় সে 180 ফিট, উপরে গিয়ে কোথায় ফেলছে জলটা নিয়ে? হি ইজ মেকিং আ পুল অফ H plus ion. এই দিকে ঢালছে, ইজ dropping the H plus ion over here, or water জলটাকে এখানে নিয়ে is making pool for the sake of understanding. Fine, হয়েছে? তাহলে এখানে যদি পুল তৈরি করে তার জন্য এনার্জি লাগবে না? লাগবে, অরিজিতে লাগবে। Where from Arijit gets this energy? Remember I have talked about when the electron flows one carrier to the other, then some of the energy comes out. This energy is required for chemiosmosis. এই এনার্জিটাকে নিয়ে what Arijit was doing was making what? A gradient of H plus ion. And this H plus ion is only allowed, Vishal bolbe, this H plus ion is only allowed to pass through F0, F1 particle. What is that called? Lock gate. The moment you open up lock gate, H plus ion এখান দিয়ে পড়বে কোথায়? On the turbine. And the turbine will start rotating. It's going to make what? Kinetic energy. And that's what is required. And it's going to, this turbine is complex 5 of electron transport system, which we call it as ATP synthase. Go over there in that page number 233 of old book, or page 160 of new book. দেখ তো F0F1 পার্টিকেলের সঙ্গে একটা ATP সিনথেজ আটকে আছে না? কি রে, F0F1 পার্টিকেল ইজ হোয়াট? ইজ আ লক গেট। লক গেটের সাথে কে আটকে আছে? ATP সিনথেজ। সো যখনই H প্লাস আয়ন আসবে, কাকে রোটেট করে দেবে? ATP সিনথেজকে অ্যাক্টিভেট করে দেবে, and it's going to make what? ATP. अंकिता बोलो, वो जो ब्लू कलर है वहां पे एक आदमी को खड़ा करो, वो अरिजीत है। बाल्टी हाथ हाथ में दे दो और देखो तो कहां से कौन से तरफ से वो पानी लेकर रख रहा है। एक तरफ क्या है, इनर सरफेस, इनर मेम्ब्रेन स्पेस है, एंड अनदर पार्ट इज मैट्रिक्स. सो व्हाट ही इज डूइंग, from he's taking the, what is this part, you tell me? This is inner membrane space and this is matrix. This is matrix. So he's making a pool over here in the inner membrane space, and that H plus ion is only allowed to pass through is through the log gate. What is the log gate? F0 particle. It is having coupled with what? ও কার সঙ্গে বিয়ে করে বসে আছে? ADP synthesis সাথে. The moment H plus ion comes in, it activates ADP synthase and makes ATP. Any issues about that? This was the discovery, and this is the statement given by Peter Michel in his experiment, and this is the process that he explained by the help of hydroelectric project. বল বুঝলাম না বুঝেছিস? Sure. Same thing, this is in mitochondria, তাই তো? The same thing you can find in the chloroplast. Go to the granum. That figure that I just now have shown you. The figure that I just now have shown you. Under chemiosmosis. See? Come on, what is this? You have maintained what? The same thing you have maintained. What you have maintained? H plus ion gradient. What is this? CF0. What is this? CF1. What is this? Log gate. And log gate is coupled with what? ATP synthase. The moment the H plus ion gets in, it activates the turbine. And turbine का काम क्या है? ATP बनाना. बोलो दोनों में क्या हो रहा है? Chemiosmosis process. This is what is chemiosmosis. So chemiosmosis में क्या बना? अंकिता, NADPH2 और या only ATP? माइटोकॉन्ड्रिया में अलग, क्लोरोप्लास्ट में अलग कुछ होता है या सेम होता है? ओनली नाम क्या डिफरेंस, एक डिफरेंस है, एक है F0 पार्टिकल और यहाँ पे क्या है? CF0, CF1. Any issues with this? Come on. বল বুঝলাম না বুঝেছি বলেছিল কেমিওসমোসিস। এর পরে গিয়ে এনসিআরটিটা একবার পড়ে, এনসিআরটিতে কি লিখেছে কেমিওসমোসিসে, এভরিথিং উইল বি, নাও ইটস গোইং টু বি ক্লিয়ার। ফাইন। সো দিস ইজ পিটার মিশেল। ঠিক আছে, চল আমরা একটুখানি দেখে নিই অ্যাবাউট লাইট রিঅ্যাকশন, যেটা বললাম সেটা একবার এখান থেকে বইয়ের কথা থেকে দেখে নিই। আচ্ছা, কেমিওসমোসিসটা দেখবো, চলো। Chemiosmotic hypothesis, what book has got to say about it? Okay, chemiosmotic hypothesis has been put forward by Peter Michel to explain the mechanism of ATP synthesis. What is synthesis? Only ATP synthesis. Where? In chloroplast and same process. Like in respiration in photosynthesis, too, ATP synthesis is linked to development of proton gradient across a membrane. So what is common in both the situations? कौन सा gradient? H plus ion gradient. And what is that called? Proton gradient. तो की maintain करा हुए चे? Proton gradient maintain करा हुए चे in both of them. Clear? Very good. Here in the chloroplast, these are membranes of thylakoid. What is the membrane? Thylakoid. In photosynthesis, protons accumulates towards the inside of the membrane, that is in the lumen. So proton कहाँ दूरी होलो? कहाँ पे वो proton आकर जम गए? In the lumen. In the lumen. In the lumen. In the lumen. This is the lumen. So the proton got concentrated where? Inside the lumen. Okay, inside the lumen. Okay. Sure. Next. Inside the lumen. In respiration, protons accumulate in the in respiration, protons accumulate in the inner membrane space of the mitochondria. Think about mitochondria. When electrons move through, ETS. NEET 2024 question. This is the line where from the question came. Okay. Cause of proton gradient across the membrane. क्या reason है proton gradient बनने के लिए? Since spreading of water molecules takes place on the inner side of the membrane. पहला, what is the reason? The reason is H plus ion gradient कहाँ पे बना? Tell me, yes. Tell me. In the lumen, this is lumen, no? This matrix are also called as lumen. P680 के लिए water got split and formed what? And this has taken place towards the matrix, towards the stroma, or towards the lumen? Towards the lumen. Water भांगलो कौन दिखे? Lumen दिखे ना matrix से stroma दिखे. Lumen दिखे. तो lumen है H plus ion बाढ़ में ना कौन बे? So what is the first reason of gradient formation inside lumen? Sir, splitting of water. बुझलाम ना बुझलाम. What is the first reason of maintenance of H plus ion high? H plus ion concentration टा बढ़ लो inside lumen. The reason की because the PS2 is towards lumen. होइछे ना है नहीं? Great. अच्छा. Okay. चलो next. This happens because the primary acceptor of electron, which is located towards the outer side of the membrane, transfers its electron not to an electron carrier but to an hydrogen carrier. এটা বুঝেছিস না বুঝিসনি? না বুঝলে জিজ্ঞেস কর. মনে হয় বুঝিসনি. এটা বুঝিসনি. This happens because the primary acceptor of electron, which is located towards the outer side of the membrane, transfers its electron not to an electron carrier but to an hydrogen carrier. Let me give this idea. Acha. A structure chalai. Come to this structure. Remember, this electron when comes back from FES during cyclic phosphorylation, FES থেকে ইলেকট্রনটা কোথায় আসে? Towards PQ. Towards PQ. Towards PQ. Calculation. What is this? PS2, PS1. Okay. What is it? Stromal, lamelli or stromal thylakoid. Having what? Only PS1. Very good. PS1. So if the wavelength of light is more than 680 nanometer, which one is going to get activated? Arjit bolbe. PS2, PS1. Only PS1. So PS1 gets activated. Kya hobe? FES. FES is going to give the electron back to PQ. This PQ is going to get this electron, and this electron will be absorbed by PQ, and PQ is going to get reduced. PQ is going to get reduced into what? PQH2. Into what? PQH2. PQH2. And this PQH2 is going to give the electron back to PS1. Fine. And this is how it is going to work. The money A PQ plus 2 quinone is going to get reduced. So who is going to accept the electron? Who is going to accept the electron? Electron carrier or hydrogen carrier? Hydrogen carrier. Got the point? Hydrogen carrier is going to take this. Hydrogen carrier is going to take this. So this electron comes back to PQ. PQ gets reduced to PQH2 and it is absorbed by hydrogen carrier. It is absorbed by hydrogen carrier. This is one of the reason. This is one of the reason why. So this happens because the 1 degree, that is primary acceptor of electron, which is located towards the outer side of the membrane, transfers its electron not to the electron acceptor but to an hydrogen carrier. So what is going to form inside the lumen? Again, a hydrogen pool, hydrogen gradient, hydrogen concentration. So two reasons we have talked about now. After splitting of water increases the H plus ion concentration. Second, the electron which comes out from the photosystem 1 is being absorbed by plastoquinone, and this plastoquinone is reduced, and this reduced hydrogen helps in increase in the gradient. increase in the gradient. Hence this molecules removes the protons from the stroma while transporting an electron. তাহলে H+ আয়নটাকে রিমুভ করে কাকে ট্রান্সপোর্ট করলো? ইলেকট্রনটাকে ট্রান্সপোর্ট করলো। দেখ কি করলাম, ওটা হাইড্রোজেনটা নিয়ে আবার ইলেকট্রন ট্রান্সপোর্ট করে দিল। When this molecule passes on its electron to the electron carrier on the inner side of the membrane, the proton is released into the inner side or the lumen side of the membrane. তার মানে বারবার যখন ইলেকট্রন ফ্লো করছে, during the electron flow, the electron carrier, what is the electron carrier? PQ. PQ is the electron carrier, and PQ gets what? It accepts the electron and it gets reduced and transfers what? The H+ ion towards the lumen. Towards the lumen. Any issues with this? Come on. Adrian, কোনো অসুবিধা আছে? Problem? Sure. Very good. Okay. Third, NADP reductase enzyme. কোন দিকে থাকে? আয়ুষী বলবে। NADP reductase enzyme present where? Inside the lumen or inside the towards the stroma or matrix? Okay. NADP reductase enzyme is located on the stroma side of the membrane. Need to know. Along with the electron that come from the acceptor of the electrons of PS1, protons are necessary for the reduction of NADP2 and NADPS2. These protons are also removed from the stroma. These protons are also removed from the stroma. Remove করে কোথায় ঢোকানো হচ্ছে? Remove করছে from stroma into the lumen. বুঝলাম? Hence, within the chloroplast, protons in the stroma decrease in number. Within the chloroplast, the H+ ion in the stroma decrease in number, while in the lumen, there is an accumulation of protons. This creates a proton gradient. দেখো, সঙ্গে আইকে নিয়ে। This creates a proton gradient. তাহলে পুল তৈরি হলো একবার, তারপরে কি করতে হবে? লক গেট খুলতে হবে যাতে কিনা H+ ion আসতে পারে, and the moment lock gate opens up, it's going to activate ATP synthase enzyme. This creates a proton gradient across the thyroid membrane as well as measurable decrease in pH in the lumen. Kire, more of H+ ion inside the lumen, the pH is going to come down or is going to go up? It's going to come down. More of H+ মানে pH is going to come down. Proton gradient is important because breakdown of this gradient leads to synthesis of ATP. The gradient is broken down due to the movement of protons across the membrane to the stroma through the transmembrane channel of the CF0 of the ATP synthase. আবার প্রোটনটা নর্মাল হয়ে যাবে কখন? When the H+ ion starts coming in through a CF0 and activating what? ATP synthase. ATP synthase activate হয়ে গেলে কি হবে? ATP তৈরি হয়ে যাবে। বা বুঝলাম, this is all about the ATP synthase enzyme consists of two parts. One called the CF0, উপরেরটা, is embedded in the thylakoid membrane. CF0 কোথায় ঢোকানো আছে? In the thylakoid membrane. দেখাবো কি রে? CF0 কার মধ্যে আছে? This CF0 is present where? Embedded inside the thylakoid membrane. CF0 is embedded inside the thylakoid membrane and CF1 is outside. CF1 is outside. Acha. And forms a transmembrane channel that carries out facilitated diffusion of protons across the membrane. Now you tell me, what is the meaning of চলছে? Okay, তাহলে facilitated diffusion. Clear? So this is what is about facilitated diffusion. That means when the diffusion is being held. Okay. Now, the other portion is called CF1 and the protrudes on the outer surface of the thylakoid membrane. On the side that faces the stroma, towards what? CF1 টা কোন দিকে আছে? Towards the stroma. The breakdown of the gradient provides enough energy to cause a conformational change in the CF1 particle of the ATP synthase, which makes the enzyme synthesis several molecules of energy packed ATP. So ultimately what happens? It makes what? There's a conformational change in the ATP synthase. Turbine is called ATP synthesis, আমাদের এক্সাম্পলে ছিল। So there will be a conformational change. যখন এই হাইড্রোজেন পাস করবে ATP সিনথেসিসের মধ্যে একটা কনফর্মেশনাল চেঞ্জ আসবে and ultimately क्या बनेगा? What is going to get formed? Clear? ATP is going to get formed. This is what is called chemiosmosis. This is what is called chemiosmosis. That chemiosmosis requires a membrane, a proton pump, a proton gradient, and ATPase enzyme, a synthesis enzyme. Energy is used to pump protons across a membrane to create a gradient, of or high concentration of protons within the thylakoid membrane, ATPase has a channel that allows division of protons across the membrane. This releases enough energy to activate ATP synthase enzyme that catalyzes the formation of ATP. Along with NADPS2, produced by the movement of the electrons, the ATP will be used immediately. ATP will be used immediately in the biosynthetic or dark reaction. The dark reaction are naam ki? Biosynthesis. Biosynthetic reaction. Okay? Or dark reaction taking place in stroma, responsible for fixing carbon dioxide and synthesis of sugar. We want to understand that what is called dark reaction. During light reaction in photosynthesis, ATP, O2 and hydrogen donor NADPS2 are formed. So what is NADPS2? It is a hydrogen donor. It donates hydrogen where? Towards stroma or towards the lumen? Ball, bujlam na, kono oshubidha ache? So that's all about chemiosmosis. That's all about chemiosmosis. So next day we are going to start with the dark reaction. Dark reaction. So in light reaction, what are the things which we have been able to make? Number one, ATP, NADPS2, oxygen, and water back recycled. This ATP and NADPS2 are required where? In the dark reaction. So next day we are going to talk about dark reaction. Kono oshubidha ache? Dark reaction er porei C3 cycle. Dark reaction is biosynthetic process. Also, it is called as C3 cycle. And after C3 cycle, on that very day, we are going to also talk about C4 cycle. And we're going to see the difference between C3 and C4. Also, what we need to do is difference between cyclic photophosphorylation and non-cyclic. ওটা দেখবো PS1, PS2 আমরা ডিফারেন্সটা একটু দেখে নেব। ঠিক আছে? এটা পাঠিয়ে দিচ্ছি তোদেরকে, পরে দেখে নিস। চলো, so next day we are going to start with the dark reaction. So that's all.